Make it right.
Table of content. Introduction, our experiment, and conclusion. Introduction. About us. Hello, we are Ayaka.
Giselle.
Anna.
We are in 8th grade, and our hobbies are to sleep. We chose this topic because we want to test something related to chemistry and food. Ripening was very interesting, so we had a try. Introduction of our experiment. The goal of this experiment is to utilize an iodine indexer to detect the conversion of plant starch to sugar in order to assess fruit ripening caused by the plant hormones ethylene. Hormones are used in plants' tissues to communicate. Hormones are molecules that are created in one place and have an effect on cells in another. The majority of the plant's hormones goes through the vascular system, but others such as ethylene are released into the gaseous phase or air. Developing plants' tissue generate and release ethylene more quickly. Growing tips of roots, flowers, injured tissues, and ripening fruit all emit ethylene. Plants are affected by the hormones in a variety of ways. One is the ripening of fruit. The starch in the fleshy section of the fruit is converted to sugar when it ripens. Then ethylene initiates the reaction in which the starch is converted into sugar. Iodine solution forms a dark-colored complex when it binds in starch but not to sugar. If you paint a fruit with an iodine solution, wait for it to darken, you can tell how ripened it is. It will be black because the unripe fruit is starchy. The more starch has been converted to sugar, the riper the fruit is. Therefore, the ripening the fruit, the less iodine compound is generated. Thus, the same fruit will be lighter.
Experiment. Our hypothesis is that storing an unripe fruit with a banana will have no effect on its ripening. The reason for this is that the banana has a low level ethylene concentration, so it won't affect the apples. Since the banana will be cut to the same size as the apples, the effect won't be present. Steps. Cut the whole apple into 4 slices, and you can keep the skin on or take it off. Cut the banana into half. Place both of those fruits in a Ziploc bag with no air for 2 to 3 days. After 2 to 3 days, get the apple slices out of the bag. Have a graduated cylinder or tray and fill it with 1 centimeter of iodine solution. Place the apple slices in the tray or cylinder and keep it there for 1 to 2 minutes, making sure that both sides of the apple are covered with iodine solution. After 1 to 2 minutes, rinse the apples out and record any observations. So as you can see here, this is what our apples look like when we had just first placed them in the bag, and this is what they look like after we dipped them in iodine solution.
Conclusion. Our hypothesis was that storing an unripe fruit with a banana will have no effect on its ripening. The reason for this is that the banana contains lower levels of ethylene compared to the apples. Since the banana will be cut into the same size as the apple slices, the effect will not be present. After completing the experiment, we found that our hypothesis was incorrect. If we had stored the apple with the banana and there was no effect on its ripening, then after placing the apple in the iodine solution, the apple would have been very dark, even after rinsing it out. Since the apples were fairly light in color, the banana clearly had an effect on the apples. If we were to do this experiment again, maybe we should change the fruits that are used. Since apples and bananas both have high levels of ethylene, the effect might not have been so great as they could have been affecting each other instead of one fruit affecting the other. Since lemons, various types of berries, cucumbers, and figs have a low ethylene concentration, these would have been better test subjects. Apricots and pears are known to have higher levels of ethylene, so pairing those fruits with these might have produced better results. The banana size also could have affected our results as we decided to cut the banana in half instead of using one full banana.